So welcome everyone. Thank you for your patience. Thank you for joining us. And it's my pleasure now to introduce Chris. Chris Langmead is Professor, Deputy Director and Better Medicine's Theme Leader of the Neuromedicines Discovery Centre at the Monash Institute of Pharmaceutical Sciences, a collaborative venture targeting new medicines development for poorly treated mental health disorders. He also co-leads the Institute's Neuroscience and Mental Health Therapeutic Program area and directs a collaborative drug discovery program with Servier. Prior to this, he was head of pharmacology at Heptares Therapeutics, a UK-based biotech company where he was responsible for all of the company's discovery biology. He is an acknowledged expert in GPCR drug discovery, particularly in the field of neuropsychiatry, where he has led multiple projects into late-stage preclinical development, many of which are now in clinical trials. Prior to joining Heptares, Chris was a neuroscience researcher at GlaxoSmithKline UK. He has a degree and PhD in pharmacology from Queen's College, Cambridge, and University College London, respectively. We are really pleased and delighted to have you, Chris. Thank you for joining us. Over to you.
Thank you very much, Scott. I do want to correct you on one thing. I've aged a lot this year, so you might not have, but I definitely have. It's really great to be able to be here at the third attempt, and my first attempt at giving a talk in the guise of our new Neuromedicines Discovery Centre, which I'll touch on briefly at the end, but I really wanted to talk about a particular research story that we've been involved in over the last 2 or 3 years. It's a collaborative venture with colleagues down at the Florida Institute of Neuroscience and Mental Health. And it is, as Scott has suggested, around developing new treatments for alcohol use disorders. Now, this is a standard slide I put at the front of many of my presentations. I don't need to go through the importance of better treatments for alcohol use disorders in front of this audience, but suffice to say that I think we would probably all find common ground if we said that alcohol use disorders are poorly addressed by current medicines. As is the case for a lot of mental health disorders, we are really quite poorly served.
Just turn it up a bit. We can't quite.
Okay.
You could go with the microphone.
Do you want me to? Sure.
Sorry.
Is that better?
Yeah.
Okay, fantastic. Sorry to everyone else now. It was like me booming in your ears. So I think, yeah, we agree that it's one of the number of mental health disorders that are poorly addressed by currently available therapeutics. So my line of work has been very much in around the discovery and development of new drugs for the treatment of psychiatric disorders. And one of the problems that we've had in the way that we treat new drug development is that often it's viewed as a linear process. We start with what we think is an interesting drug target. We try to identify new compounds, new chemicals that might interact with that drug target. We test them in animal models. We manufacture what we think is our best medicine, and then we take them into healthy volunteers and then our patient populations with the hope of getting a new approved drug for that disorder. The trouble is that quite often where we fall down is at this stage, where we take all of the work that we've done in the early stages of the drug discovery and development process, and we move towards our clinical population, our patients that we care about. And unfortunately, we find that essentially our hypothesis doesn't work out. The approach that we've taken doesn't bring clinical benefit to the patients that we're seeking to treat. So one of the ways that we've tried to adapt to many of these problems, and this is particularly a problem in neuropsychiatry, but it's by far not just a problem of neuropsychiatry, it's a problem for drug discovery and development more generally. But one of the approaches that we've taken is instead of thinking of this as a linear process, why don't we think about it as a cyclical process? So let's start right at the top with our patients and use the information, as much information as we can from understanding the patient population, and then use that information to drive the identification of new approaches to treating these mental health disorders, use that information to identify potential new drug targets and identify new chemicals that interact with them, and make sure that the animal models that we're then using actually align with the patients. They're predictive of what happens in a patient, because if they're not, then we're essentially wasting our time, we're wasting our money, and we're not serving people like patients and yourselves as best we can. So this is the approach we took in this project around alcohol use disorder. And it's here now that I have to credit my friend and colleague, Andy Lawrence, who works down at the Florey Institute, and some of you may well know him. He's an excellent alcohol and addiction researcher. As part of this project, we started with some post-mortem samples taken from alcohol use disorder patients that were non-smokers. So we have a very clear, well-defined population of heavy post-mortem tissue from heavy drinking patients, but non-smokers essentially. So they're not addicted to nicotine, but they are specifically addicted to alcohol. And these two groups, we have a control group essentially, who don't really drink, and an alcohol use disorder group. And I'll show you the characteristics here. The two groups are age matched, they're BMI matched, but you can see in terms of the quantity of drinks per week or alcohol intake, these alcohol use disorder patients were drinking very heavily at the time of their death. And what we were particularly interested in is understanding what happens in the brains of those patients that differentiates an alcohol use disorder patient from essentially a control, from a normal sort of healthy human being. And we were particularly interested in two regions of the brain known as the caudate and the putamen. So these are subcortical regions. And without going into too much detail, the caudate is responsible for what we would probably describe as goal-directed behavior, essentially voluntary behavior. You choose to perform an action. Whereas the putamen is responsible for habitual behaviors, things that become habits, and over which you start to lose control, essentially, because they become habitual. And there is a quite well-described hypothesis in the literature that during states of addiction, including alcohol addiction, that control over these behaviors around drinking or opioid use or methamphetamine use, control over that behavior moves from the caudate to the putamen. So essentially it moves from a goal-directed voluntary behavior to a habitual behavior. And then there might be a molecular underpinning for this process, and that's what we set out to look for. So we used we had 40 brain samples, and we did a high throughput RNA sequencing approach to look for the expression of different genes across these tissues in our control and alcohol use disorder patient population. And as you would expect, we looked at around 14,000 genes. 13,500 of these genes are unchanged. Nothing happens. However, in a very small number of the genes, we see either they are significantly upregulated in their expression in patients with alcohol use disorder, or they're significantly downregulated. And these are the ones that we start to hone in on, because these are the ones that we're potentially interested in. Are these genes and the proteins that they encode for potentially involved in making patients susceptible for that transition into alcohol addiction? Oh, I've lost me bars. Anyway, it's fine. Because you're not going to be interested in the gene names anyway. But suffice to say that in this graph, there are a number of genes that we identified that either go up in their expression or down in their expression. And for me, as a pharmacologist, I'm particularly interested because a number of these genes encode for what we call G protein-coupled receptors. Now, many of you probably will have studied pharmacology in the past and have blocked this part of your education out of your psyche because of the trauma that you suffered when you learned the pharmacology. So I'll promise to make this not too traumatic for you. But just to remind everyone that G protein-coupled receptors are the largest class of targets of FDA-approved drugs. So around 30 to 40%, depending on how you count it, of FDA-approved drugs act via G protein coupled receptors. And you'd be very familiar with them in terms of beta blockers and antihistamines. You'd probably be familiar with the nature that naltrexone exerts its effects through G protein coupled receptors, in this case, an opioid receptor. And for those of you who are particularly interested, this is what a G protein coupled receptor looks like. It sits in a cell membrane. It has 7 transmembrane spanning domains, and basically it mediates the actions of all different kinds of hormones, neurotransmitters, etc. Over outside the cell, like outside a neuron in the brain, changes the shape and then transmits that signal into the cell through the actions of intracellular binding proteins that you can see in the blue and the orangey red and yellow. So this is the family of receptors that I'm most interested in drug targets. I have a lot of experience in working with these. So I was particularly excited with Andy when Andy showed me the data and said, look, we have all of these GPCRs that change in alcohol use disorder, specifically non-smoking alcohol use disorder. And one of the ones that we were particularly interested in was a receptor called the muscarinic acetylcholine M4 receptor. And it's a G protein coupled receptor. It mediates the effects of acetylcholine, which you are familiar with from the cholinergic system, the parasympathetic nervous system. And it's found mainly in the brain. So it's not expressed in the periphery to any great extent. It's actually fairly well concentrated in the brain. So we dug a little bit deeper into some of these data and looked at the expression of, to confirm the expression of some of my favorite G protein coupled receptors. You know, we're all sad and we all have our favorite receptors. And so I'm looking at the cannabinoid receptors and dopamine receptors and CHRM4, which is the muscarinic M4 receptor. None of these receptors change their expression in the caudate. So this is the caudate. This is responsible for your voluntary actions, okay? However, when you look at in the putamen, the M4 receptor, amongst many others, is significantly downregulated. The levels of expression go down. And we start to imagine, well, is this part of the disease process? Is this something that makes patients more susceptible to drinking? And we confirmed this is just at the gene expression. And we actually confirmed that selective downregulation of the M4 receptor was found at a protein level as well. So it's not just a gene artifact. We're actually now looking at the protein in the human tissue, and we see that that level of expression is down in the putamen, but not in the caudate. Great, very interesting and exciting. But what happens? How do we use this information? Well, we have to go back now and think about, well, how do we actually start to develop this preclinically? What do we need to do in order to think about this as a bona fide therapeutic target for alcohol use disorder? And that goes back to the animal models and the preclinical models. So how do we do that? How do we model alcohol use disorder preclinically? In this case, in a strain of rats known as IP rats. Well, we put them in a chamber and we give them a choice. They have a choice, what's called a 2-bottle choice paradigm, and they have a choice whether to drink water or 20% ethanol. And they choose 20% ethanol, this strain of rats. So over a long period of time, they become essentially addicted to and dependent on alcohol. And we do this chronically. There's no point just doing it for a week. Doing it for a week does not mimic the chronic drinking that you will see in the patients that you work with. So we do this for 2 months, 4 months, 6 months, sometimes 12 months. They're long experiments. But we want to, as best we can, recapitulate what you're seeing in patients. And this is all wonderful work by a postdoctoral fellow at the Florey named Lee Walker. So to cut a long story short, because there's lots of colors and lots of bars on this graph, to cut a long story short, we see the same thing happening in the brains in rats exposed to long-term alcohol as we do in patients. Rats don't have caudates and putamens, but they have similar brain regions, analogous brain regions called the dorsomedial and the dorsolateral striatum. And we don't see any changes in expression of these proteins in the dorsomedial striatum, like the caudate, but we do in the dorsolateral striatum, which is like the putamen. So now this is really important because we're seeing a preservation of effect between what we saw in the patient samples and what we're now seeing in our models in which we would like to potentially test our new medicines. So that's another tick. We've potentially got continuity in terms of our animal model. That's great. I've got to target the muscarinic M4 receptor. How am I gonna investigate that? How am I gonna probe that? And we're going to do that using a tool molecule, which I've just called VU154. And VU154 is a special kind of drug that actually potentiates the function of the M4 receptor. Now, I did have a bunch of slides that showed you how it does this at a molecular level and all the pharmacological analysis and stuff. But then I realized the best way to describe it is think of it like a dimmer switch. In this case, it's dialing up the activity of the muscarinic M4 receptor. It makes it more sensitive to acetylcholine, so essentially it boosts its function. That's what it does. And if you remember, we were dealing with a situation in our chronic alcohol-treated animals or our patients suffering from alcohol use disorder, where we think the muscarinic M4 receptor is downregulated. So we want to bring it back up to where it was before. And we can do that using this tool compound, which was courtesy of some outstanding work done by colleagues at the Vanderbilt Center for Neuroscience Drug Discovery in the USA. So now what we do is we bring all of these things together as a proof of concept. So this is how we model the alcohol addiction and test these compounds in the rats. So rats go into an operant chamber. These are rats that have already been long-term drinking on that 2-bottle choice paradigm. So they're already very keen to drink 20% ethanol over water. And we still present them with a choice. We present them with an active lever, which gives them alcohol, and an inactive lever, which gives them water. And associated with the active lever, we have a light and a smell, okay? And they start to associate the light and the smell with the alcohol lever and the delivery of their little shot of 20% ethanol. And in this case, we did this, I think, for 60 or 90 days, long-term exposure. And then we treat these animals with either our M4 potentiator, the VU154, or just vehicle control. And we measure how much alcohol they consume. And then 7 days later, we switch it around. So the animals that had vehicle before get the treatment, and the animals that had the treatment before get the vehicle. So it's all counterbalanced. So every animal gets every treatment. So that's great. That's going to tell us about how much does an animal want to consume alcohol, which is obviously part of what we need to battle with in patients suffering from alcohol use disorder. But obviously, we need to think a little bit more about relapse as well. And this was a slide that my student put together at the time. And because she liked going to beer club on a Friday, she would go to beer club until I made her write a thesis, and then she couldn't go to beer club anymore. But unfortunately, when it came to crunch time, very stressful, placing her in a very stressful situation turned her to beer. And I make a joke of it, but it's not. We know that patients placed in either stressful contexts or in with cues around them, like in a bar or something like that, are more susceptible to relapse to drinking, even if they've not been. So that's a really important part of what we need to measure. It's not just drinking, it's relapse to drinking as well. So we can model that as well. So we have the same paradigm. We have our rats sitting in our operant chamber. They've got a choice between alcohol and water. And then we extinguish them. Basically, we switch off the choice of, which take away the water, we take away the alcohol, we take away the lights and the smell, and basically they come off that paradigm for 7 days. So they're extinguished and they actually stop drinking in that time, generally, or they significantly reduce their drinking. And then we treat them with either the drug or the vehicle. And then we put them back in the chamber. And this is where we do something which I think is quite clever. So we don't allow them to drink, they don't get water, they don't get alcohol, but they do get the light and the smell. That's their cue. And then we measure how many times they press the lever for the alcohol, because they've got the smell and they've got the light. And basically, this is what we call cue-induced reinstatement. They've got all the cues to be drinking, and we want to see whether they do, and to what extent they do, and whether the drug can change that. And then basically, we do the whole thing again. Each animal gets every treatment. So these are fairly long and onerous experiments, but when you get good results, they're worth it in the end. So these are the data with the M4 compound when we dose it systemically. So we give it by mouth. A dose of the VU compound significantly reduces the amount of ethanol, alcohol, that these rats will self-administer. It cuts it about in half, which is a nice result. What's even more encouraging is that when we do the reinstatement test, so you can see here in blue the extinction. This is what the animals are drinking after we've taken off alcohol. As soon as we do the reinstatement, as soon as we present them with the light and the smell again, they start banging away on the lever because they think they're going to get alcohol. And if we do that in the presence of our drug treatment, again, that cuts it pretty much in half. Now, one of the things we always want to be careful about here is, well, are you really affecting alcohol addiction, or are you just taking away someone's drive for anything fun in their life? Okay, so there are a number of ways we can look at that, but one of the things we can do is to see how specific it is, and it is pretty specific in this case. So we measure, for example, sucrose self-administration. Are these animals still keen to consume sugar? And yes, they are. The drug doesn't change that, but it does specifically target the consumption of alcohol. And we did quite a nice little proof of concept at the end as well. Said, well, is our hypothesis correct? We had this hypothesis that there was this one teeny tiny region of the brain that was involved in this process. So what if we injected the drug directly into that region of the brain using a micro-injection, a tiny, tiny, tiny volume, but it's not going to go anywhere else in the brain? Well, the good thing is we pretty much see the same result. We see a decrease in the drive to consume, self-administer alcohol, and actually probably even more marked depression in the propensity to relapse, to essentially trying to consume alcohol in this model. So these were really fantastic data for us. It was a terrific proof of concept, but proof of concepts for basic scientists don't help people like you, and it don't really help patients in the short term. So where are we at in a therapeutic context? Well, there are drugs with this mechanism currently in clinical trials. Now, they're not in clinical trials for addiction, but they are in clinical trials for other disorders. And one drug is this ex-Pfizer drug, which is now being progressed by a company called Cerevel. They're progressing it as a treatment for schizophrenia. They recently had some very positive data in a phase 1b clinical trial. So this molecule will be going into phase 2. What I'm trying to say here is that there is the possibility that not too far in the future, there will be an approved drug with this mechanism of action. So the potential for treatments to be available for alcohol addiction and potentially other addictions as well is not perhaps quite as far away as we might think, just from the sort of basic science, as it were. This has opened up a whole new range of different possibilities. That's all I wanted to talk about for M4. I just wanted to finish up with 2 or 3 slides to talk about something that's come to light since the first time that I was invited to speak here, and the second time, actually, which is the opening of our Neuromedicines Discovery Center. And this is a collaborative venture between Monash and all of the partners that you will see on the slide here, the Florey, other partners, the Department of Psychiatry, the University of Melbourne, Behaviour Works, the Phoenix, Australia, the Treatment for Post-Traumatic Stress Disorder, and the Melbourne Neuropsychiatry Centre. And we are very interested in developing a whole range of different approaches, including the one that I've just described to you about treatment of mental health disorders. But one of the first things that we are actually looking at, in the context of some of these difficult to treat mental health disorders, some of the recent data that's come out, and you may be aware of, with the use of psychedelic agents in the treatment of difficult, challenging mental health disorders. And I'm sure that you will probably be aware of the approval of intranasal ketamine for the treatment of depression. Some recent data, and actually I should have updated this slide because there's now some other trial data come out on the use of psilocybin in the treatment of depression, and the use of MDMA in the treatment of post-traumatic stress disorder. So the model for the use of these agents is that you only need to give 1 or 2 doses of these compounds in a clinically controlled setting as an adjunct to psychological therapy. And essentially, it's that regime with a sort of a pre-session and a post-session as well of psychological therapy that seems to produce quite long-lasting clinical efficacy in a number of different mental health disorders. And we're really still so much at the beginning of this. And the center has been set up such that we not only are going to do some clinical trials in this space. So this is what we call our Better Minds. You've always got to have names for these things. So Better Minds is our clinical trials arena. And we're going to be doing some clinical studies to look at some of the effect of some of these medicines in different mental health disorders, including hopefully addiction. But we want to understand better the mechanisms of action at play as part of our development of next generation therapeutics as part of our better medicine space. And our better futures is very much around how do we take some of these findings and actually move them into healthcare practice with people such as yourselves. Because it's one thing to have interesting clinical findings, it's another thing to translate that into meaningful clinical outcomes in a population at large. And I want to leave you with one final slide in this space, which was some data that I only became aware of a couple of weeks ago, which was published by Ben Sasser in the UK, using MDMA-assisted psychotherapy as a treatment for alcohol use disorder. It's published in Pharmaceutical. So it's a very small-scale trial. I can't remember the number of patients, but I think if you look at the graph, you can see here, taking these heavy drinking patients and then giving them a regimen which is essentially detox plus treatment as normal, as they would normally treat them, versus a detox and 2 sessions of MDMA-assisted psychotherapy. And I think you'll see there's quite a stark difference in terms of the number of patients that return to heavy drinking after that treatment regimen. And interestingly, to see that how long-lasting an acute treatment can be in this space. And we're on a precipice of something very interesting here, and that's what we want to explore over the next couple of years. So with that, I'm going to thank everybody that was involved in the work, colleagues at Monash, at the Florey, at Vanderbilt, support of NHMRC, and the UNSW, and obviously support from the Australian government as well. Thank you very much.
Thanks, Chris. Well done, really appreciate it. So maybe there are some questions for Chris on that presentation. I'll kick off if I may, and that is, where do you see the research, and where do you see things in 1 year, 3 years, 5 years? What's the pathway? Where will you be if we invited you back in 5 years?
I think you will. I think you'll see that we'll have a lot more data in terms of the use of. I'm going to start with the psychedelics and work backwards. You have a lot more data in terms of the use of things like MDMA in alcohol use disorder, opioid use disorder. There's going to be a lot of need for well-controlled clinical studies to tease out the potential efficacy of these approaches across different mental health disorders. And there are lots of people doing this. There's lots and lots of activity. So I think in 5 years, 3 or 5 years' time, we'll have a huge data set in that regard, and we'll have a number of different potential treatment options. It's not going to treat everyone. It's not a magic bullet, which is why the other approaches that we've been taking, where we really seek to understand what's happening at a molecular level in patients to become addicted, how can we target that process? How can we reverse that process? I think we'll start to see in the next 5, 6, 7, 8 years, some of those newer medicines becoming available and going to clinical development. It's challenging, but someone's got to do it.
Any questions? I take it, as I go over to Daniel, one of the challenges that you have is you can't offer mice talking therapies.
Well, you can try. No.
So while I understand that sort of the administration of MDMA, for example, is done in conjunction with talking therapies and therapy sessions, I wonder about that.
We can't model that in animals. We can only model what happens in the brain to understand what an MDMA or a psilocybin or a DMT might do that changes the brain and makes it more receptive to those kind of therapies.
My question was just regarding the treatment for schizophrenia that you talked about that was targeting the same. I'm assuming the group that they're researching, a substance use disorder as well as schizophrenia, would exclude them from that study, or? I
imagine they probably would, yes, because knowing how phase 2 trials are designed, they're very tightly designed with lots of different exclusion criteria. I think the hope is if you haven't got companies that are willing to take that on themselves, then either they'll be willing to sub-license people to take that on in a specific population. Now, my colleague Andy at the Florey has taken a Merck drug that is approved for insomnia, which is Belsomra, Suvorexant. And he is running, or has run, I can't remember where it's at, a clinical study to look at that for essentially comorbid sleep disorders and alcohol use disorders. So he's doing that under license. And I think there are opportunities to do that. Otherwise, we're going to have to wait for drugs to be approved and then use them off label until such time as, you know, we can show that they are effective. And that's a slow and torturous route. I don't really want to go down that route. Big pharma companies are not keen on doing trials in addiction. I'm sure we all know that because they're challenging. I think that's why it's incumbent on researchers, practitioners, and commercial entities to work together to find a path forward, the fastest path forward in this space. These treatments that we're all using, they're all based on science that's 50 years old. It's rubbish.
Yes, Chris, I've actually got one question from our Zoom attendees. It's from Michelle Kethley, and it probably connects to the other question, which was in terms of the studies that you've mentioned, are they looking to refer clients? Is there a way to refer different clients or individuals or for recruitment to the study? To the psychedelic studies, I assume so. I'm going to answer the question on that basis, and if not, correct me. There are going to be a number of clinical trials in this space. There already are clinical trials in Australia recruiting. St Vincent's has some, Monash University have some. The federal government announced early this year $15 million to support clinical trials in this space. And I don't think, you know, there are going to be some trials that will be funded. I think I know of 4 trials that will be funded in this space, in that space, basically. So you will see in the next couple of years a substantial increase in the number of clinical trials in Australia across, and I know at least one of them is an alcohol use disorder. I think there are more, but I know at least one of them is an alcohol use disorder specifically. Excellent. Thank you on that. And just to confirm, will the slide presentations, are you happy to share those with attendees today? I will. Excellent. Thank you.
That's great, Chris. Really appreciate that. Thank you. I mean, one comment maybe rather than a question is more about the appetite for novel treatments, because it strikes me that the science is one thing and the evidence is one thing, but it also requires the timing has to be right for governments and others, including pharmacological companies, to pursue these things. And I think with the MDMA and the treatment that's just emerged mostly this year, it's really felt like there's a whole lot of momentum's come behind that just in the last 6 months or so, in part perhaps driven by the veteran community. But there is at the moment in that space quite a surge of interest and activity. Could you comment on not just the science leading and the evidence leading things, but how you have to manage, in some ways, the other factors that enable drugs to get up and over the line.
So 2 comments, one commercial, one sort of societal. Commercially, you're right, actually, a whole bunch of companies stopped working in psychiatry. They put it in the too hard basket. They said, we'd rather work on oncology, we'd rather work on cardiovascular disease, because quite frankly, it's easier. It's still not easy, but it's easier. We are now seeing inevitably a regression to the mean. We're getting a bounce back. There's actually quite a lot going on in the neuropsychiatry space now, and it's really becoming more interesting. So I remain hopeful, and I will keep pushing it until I retire and probably beyond, that we keep trying to develop new, better medicinal approaches for mental health therapeutics. In terms of a sort of societal and political perspective, you're right. The appetite needs to be there. There's been a lot of work being done in the background. I think the use of medicinal cannabis has sort of paved a pathway for this. And I think people are in a much more receptive frame of mind. And then you present them with the evidence and they become even more receptive. So I think I think that is changing. I mean, the fact that the current federal government, and we know which way they lean politically, was willing to put up $15 million in support of clinical research in this space, I think speaks to the way that the landscape is changing.
Well done. Thank you, Chris. Please thank Chris Langmead for joining us today. All right, we'll take a short break now, and then our next presentation is with Dr. Nico Clark and the use of long-acting injectable buprenorphine for the treatment of opioid dependence in the medically supervised injecting room. We'll be back shortly.